Hello, everybody. I hope everyone's having a nice and safe snow day. In order to try to keep on track, I am going to do a quick recorded lecture for today where we're going to finish up the Mechanistics of Toxicology chapter. Make sure that you guys are keeping up with the readings on the book. Also, don't forget that on Wednesday is when your long-term research project topics are due. The first one is basically just the topic paper, where you guys will tell me what your question is, what the topic toxin is. You also should give me a very brief description of it. I do not need you guys to give me lots and lots of details. You don't need formal references at this point. You essentially just need to be able to tell me what you are working on and your general idea. So I always kind of think of this as like your Wikipedia level of diving into the subject for it. So that'll be due on Wednesday. So make sure that you guys are focusing on that. So jumping into the material that we had covered previously. All right, jumping back into where we had left off. We had already gotten through that step 0, the idea of delivery to the body. And that step 0 delivery is essentially getting the toxin from the source to your body. So for example, if it's an airborne toxin that's being released from the tailpipe of a car, step 0 is how does that drift towards you to actually interact? And then exposure, step 1, is the dosing, getting it into your body, like by inhalation, insufflation, by cuticle transport, as well as, remember, it's the transport to the target cells. Now, this is essentially the fate and flow throughout the body. Remember that depending upon how you're dosed, the amount of capillaries, the thinness of the membranes, certain material are going to move faster into the body and also out of it. But once you have your dose in your body, its ultimate fates are either to be excreted and cleared from some part of your body. You also can have the target's cells receive the toxin versus it going to a non-target cell. So for example, let's think about a hepatotoxin, something that's going to affect your liver. If your liver is the target, and let's say you've been bitten on the leg by a snake, it's going to get transported up, pushed down, pushed out through your various blood vessels, and some of the poison will end up in your liver, while others are going to end up in non-target organs. So let's say some of it ends up building up in your thumb. It's not going to affect it. So really, we're thinking about taking that dose and then dividing it out by the total body mass. This is why when we think about dosing, we are always focusing a lot on the micrograms of the toxin per kilogram of body weight, because this basic chance of it delivering to the target organ or not depends upon body size. Now, the last kind of fate is sequestration. That's where it's going to end up building up in some tissues that may permanently bind there or may get released a little bit later. So for example, fat-soluble toxins can build up in adipose and fat tissue and slowly get released over the years. Certain heavy metals will end up getting built up into your bones. Others can get put into tissues like in your hair, which is why hair follicle testing for certain metabolites for drugs and poisons can be useful. The last step of exposure is getting to the cells and either entering the cells or interacting with the surface molecules on the cell itself. Now, with the interaction, we have to think about getting through the cells. Now, certain cells are going to be more reactive than others. Any cells that are highly metabolically active, that have thin membranes and are constantly moving things in and across the membranes, are going to be really reactive. This is one of the reasons why we have special toxins that work on neurons, because neurons are heavily metabolically active. They're also quite thin in their membranes, so they are more open to having toxins affect them, versus other cells that are not as active. So for example, let's go back to the idea of adipose tissue. Yes, it is moving materials in and out, but not that often, and it doesn't have a very high metabolic rate. Now, once you get to the targets, we have to kind of think about a series of potential outcomes. Now, if the target molecule is something like an enzyme or a channel protein, you can have effects that can cause allosteric changes. Now remember, the concept of allo is different, steric is shape. So you're effectively changing the shape of the molecule. So for example, if the target is an enzyme, where nominally it's got its active site where its substrate can fit in, form the enzyme substrate complex, the molecule works. But we could have a toxin that acts as a competitive inhibitor or a non-competitive inhibitor. Remember, competitive inhibitors are ones where they land in the active site and physically block your substrate from binding. A non-competitive inhibitor is more like this example, where you have a secondary spot which changes the shape of the active site, preventing the substrate from forming. Now, the enzyme substrate complex may still form, but at a lower rate. So we can have inactivation. We also can have activation, where an active site that is nominally off gets turned on. Now, this also works for transporters and other channel proteins. Anytime that we have the channel proteins, we can block them up like competitive inhibition. We also have the ability to effectively have them get inactivated or opened by allosteric changes. So just think, changing the shape changes the function, enhances it or shuts it down. We also have hydrogen abstraction. This is the idea of taking a proton or a full hydrogen out of the molecule, binding it to the toxin, potentially permanently or temporarily, that then can form a free radical. Now, free radicals are highly charged molecules that are able to absorb and transport protons or electrons. Now, we focus a lot on reactive oxygen species that love to pull in electrons because they have the ability to essentially grab electrons from another molecule, cause damage, and then release them, causing even more. I always think of this as like a wild cannonball smashing around in your cells, doing tons of damage. So these issues with just general redox damage from pulling out protons, pulling in electrons, can cause a lot of damage. Now, we also can have the toxins act as enzymes directly, where they are proteins or protein-like substances that act as catalysts that enhance, change, mess with the metabolic system. Now, with the toxins, do remember that there are going to be direct toxins that would directly affect the target molecule, like in the top example, acting as an inhibitor. You also are going to have ones that act through a secondary mechanism. So for example, cholera. Cholera is a toxin that is produced by a bacterium which is associated with unsanitary conditions, essentially. So this cholera toxin lands on a surface receptor that then creates a secondary messenger that is mediated by a G protein system that then creates the cyclic AMP that activates protein formation that then encourages the release of chlorine and sodium into your gut. So it's this pretty complicated thing, but it's essentially doing that allosteric changes, activating channels to encourage them to produce more, just now by a secondary effect. Now, the way that this toxin kills you isn't directly. It's actually by this long chain that then creates the movement of chlorine and sodium that then pulls all the water out of the interstitial space of your gut lining into your gut, giving you horrible diarrhea, and you run out of water and potentially die that way. So it doesn't have to directly affect it. It could be an indirect effect of things. Now, once we actually start thinking about other options for how we're going to damage the molecules, we can have those changes in the rates. We can also have just general dysfunction. Think about alteration of the DNA, mutations cramming into them. We also can have changes to the enzymes, where the enzyme is no longer doing the same thing. So for example, some poisons will change an enzyme that is nominally part of your metabolic system that's supposed to help break down your food, but instead of producing metabolites that we can feed into like the Krebs cycle, it's going to produce reactive oxygen species that could cause other damage and cause bigger issues within your cells. Now, we also can just have good old-fashioned destruction, breaking down the molecules into its constituent parts. So like this cell is being affected by a destructive toxin that is causing all of the cellular machinery to essentially break down, and it's going through a process I'm going to go through in a second called apoptosis to try to contain all of these broken down points. Now, the last way that we're usually going to have interactions is neoantigens. Neo is always going to be new. Antigen is going to be associated with your immune response. So something acting like an antibody or creating a foreign protein that causes your body to attack itself. Think allergies. Think sensitization. Classic example, think about that urushiol oil in poison ivy, creating that immune response. So the damage is actually being done by your body itself, not by the cells or the toxin interacting directly with your cells, but by creating a secondary metabolite that effectively is then damaging your cells. I do want to spend a little bit more time on ROSs, reactive oxidative species. These guys are going to be based on compounds like sulfur. So like SOD2 is actually going to generate a reactive oxygen species of like sulfur dioxide. They're also going to be ones that are based on nitrogen. So just think of these as molecules that contain oxygen, like NOx's, SOx's, that are very electron grabby. They're able to grab electrons from another molecule. And when they abstract them, a lot of times then they can donate them to somewhere else. So what this is doing is just randomly destabilizing molecules, which can have a major effect on all cellular function. Now, for example, let's just think about starting with the genes. Reactive oxygen species can end up damaging the genome directly, which can end up with changes in gene expression. They can also be secondary methylation that then causes transcription errors. You also can get issues with your telomere shorting, basically shortening the lifespan of your cell's ability to replicate and repair. Now, of course, if you change the genes, then you change the proteins, and then you can have massive cellular dysfunction. But you can also have effects throughout every system on the cell. So, for example, ROSs can then damage proteins directly, leading to protein degradation or enzymes not functioning correctly. If we jumped into the good old mitochondria, where we've got our outer membrane or inner membrane with things like our electron transport chain being bound into the matrix membrane, where normally this is used for processing energy and our use of the proton motive force and ATP synthase and all that stuff you probably remember from 151. But the toxins can do a lot of damage, remodel our cytochromes so they're less efficient at pumping protons. Or even worse, just take out a cytochrome completely and end up reducing the amount of energy. You also can have effects on the function within the mitochondria. Because remember, mitochondria have their own DNA. They are endosymbionts living within each of your cells. So effectively, you kill the mitochondria, you kill the host organism because they run out of damage. So we worry a lot about ROSs because they do so much damage. But this is why antioxidants are built into every one of your cells. Compounds like carotenoids that can grab on and neutralize reactive oxygen species so that we don't have to worry too much about the damage being done. So, you may freak out, but you've got probably 10,000 ROSs being created by accident just as your body is processing proteins. It might make an NOx instead of making urea like it's supposed to. It just happens. But your body is able to detoxify it, but with certain toxins enhance ROS generation, so much so that it overwhelms and basically swamps the body's defense mechanism, leading toward all of these negative issues that we have. Now, jumping on to the next step, we've already damaged the target molecule, so now we have to kind of take a step back and go, what's going to happen with the cell itself? So it's going to depend upon the different target type of cells and what the target cell's job is. And what the ultimate effect is really kind of depends on that job and how badly is it affected. So for example, one of the classic ways that cells go into dysfunction is with synthesis errors. So think of cell division, carcinogenesis, wild cell division, or the cells not replicating. There are toxins that prevent cell division, so you're not able to repair yourself. You also can have transcription errors where effectively you are not able to make a good transcription, make your mRNA correctly, as well as issues with translation and protein synthesis directly. So just think of the DNA and protein synthesis as being very negatively affected, which is of course going to lead to cell dysfunction. You also can have failures in regulation. Now think of regulation as the body's own ability to maintain homeostasis and adjust to conditions, hormetic changes. Now, it is very common to have regulation be altered by toxins that are going to affect channel proteins, especially on your nerves. So for example, your nerves have a series of receptors for chemically gated channels that allow them to either be encouraged to fire off or discouraged. Remember all that EPSP, IPSP type of things that you've probably seen in other classes. Certain toxins and medications will affect the polarization. So for example, what we've got here is we have the resting potential of a nerve cell. And your nerve cell should be somewhere between -70, -90 millivolts at resting. For this rat, it's sitting at a beautiful -77. Now, as we get closer to the hypothetical threshold, you're going to have the nerve cell fire off. Now, in this case, we expose the rat to dexamphetamine, a type of amphetamine. And then within 1 minute, you have this rapid rise in the cell potential. We get closer to zero, we're getting easier to fire off. Thus, the effect of the medication is to make nerves easier to fire off, speeding things up, which is why amphetamine is speeds up the responses. You also can have issues with what's known as endocrine disruption. This is where a compound appears like a hormone and can have that effect on the body. Now, this can be really useful for medications. So for example, one of the very popular medications now is previously used for type 2 diabetes mellitus, but is now used as a weight loss drug, Wegovy. All these GLP-2 semaglutide-like agonists, where what they do is they essentially prevent the uptake and alter the production of secondary hormones that then encourage you to feel more full. You also can have different compounds that act as hormones directly. So, for example, PBA. People are worried about bisphenol A because it is a estrogen mimic. It looks like to your body like estrogen. Now, for a lot of you guys, this doesn't really matter too much. If you get bisphenol A, which is found in certain things like plastics, and you drink out of a water bottle that's got a little bisphenol in it, not a big deal. And even males, you guys are not going to get affected by very much at all. Now, for one thing, the NOAL level is really, really high for this. You guys would have to drink water that has been boiled for about 24 hours to get as much bisphenol A out of that water bottle as possible, and then drink about 11 gallons per day to get to your NOAL level. Now, realization that's not a big deal, but if you turn your water bottle over now, you're probably going to see like a marking or a sticker that says, BPA free on it. This is because people freaked out because since it's an estrogen mimic, in developing males, it can lead toward changes in male developmental processes, leading toward the potential for reduced male genital size. And if you ever want Congress or other organizations to jump on things, have stuff that threatens males and masculinity. You'll see them move really, really quickly to ban things. Now again, the NOAL level is so high that you don't have to worry about it, but there are other medications and things where the NOAL is low enough that we can use them for their effects. GLP-2, pills, the birth control pill, I'll show you that more in a second. Now you can also have changes to internal maintenance. Changing ATP generation. Just think about the mitochondria example that I showed you in the last one. Also messing with calcium balance. Calcium is used in your body for a number of different things, including triggering your muscles to fire off, triggering your nerves to fire off, moving calcium across the membrane in places like your lungs and your other mucous membranes to move water. So messing with calcium balance, you can really mess with an organism. Now, you also can damage membrane integrity directly, where you make the membrane more or less transmittable and changes selective permeability. So for example, there are some compounds which will cause the membranes of your mitochondria to become leaky to protons. So instead of the protons going through ATP synthase and spinning your cute little ATP synthases, giving you ATP, it is going to generate no charge. It'll just slide back into the matrix and then get pumped back out. Now, this sounds kind of cool if you think of it from a diet drug standpoint. And people come up with this every once in a while as we could use compounds like these as diet drugs because, well, if they're leaking back through the membrane, the protons aren't generating ATP, you're getting less energy out of the food you're eating. Less energy out of the food you're eating, you have to use more of the available food, and you don't store it, and you pull out of your fat stores. Sounds great, except for remember there's always secondary effects, especially when we're dealing with maintenance combined with regulation. The body doesn't know what's happening, so it tries to fight it based upon like historical context that's developed through evolution. So like the response to this drug is to think that these cells have been infected by a virus or bacterial toxin or something like that. So the body ramps up temperature in order to try to enhance metabolism and fight the invaders. Now this is a bad thing because when you ramp up the temperature, it then encourages more leaking of the membranes that then encourages higher temperature, and people essentially die by getting cooked alive by their own cells running out of control, all because you messed with membrane integrity. Now, you can also have more total body external maintenance issues. So integument loss, basically holes in the skin. We'll see this in a bit with chrome holes, where you can have the outer skin essentially get eaten through. You also can have what's referred to as loss of systemic control. Think of this as the body's inability to maintain normal balance and function and things like that. So think of the damage due to acute intoxication from ethanol. Someone drinks a lot of ethanol, they lose their systemic control, their balance, things like that. And one of the ways that people end up dying is, especially on icy days like today, people walk out of the bar, they slip on the ice because their balance is off because of loss of systemic control, and they fall backwards, hit their head, and that's all she wrote. So it can be an indirect effect, or it can be just systemic control of things like your kidneys are no longer able to balance. So just think of this as large scale effects too. Now, a little bit more on that idea of signal transmission. Those endocrine mimics are going to look a lot like the regular hormones, just not be exactly the same. So for example, estradiol, ethyl estradiol, these guys are used in birth control pills because they look a lot like estrogen and work on the same pathways of estrogen and progesterone, convincing the body that it is already in an pregnant state, so you do not have secondary pregnancies occur. So effectively, it's in a pretty good birth control. And the nice thing is, this does not develop a tolerance. Certain others do. And remember, tolerance is the more that you are exposed to it, the less the body is reactive. So for example, this is an image out of a paper looking at the hypothetical effects and what is being activated, and these are all different genes, don't worry too much about all the names, that are causing both positive and negative effects on the nerves and exposure to nicotine. Now, in a nicotine naive state, so an organism that has not been exposed to nicotine before, What happens when you give them an acute dose of nicotine is you have excitability and you have genes being activated that essentially causes the nerves to fire off. It's all the beneficial aspects of nicotine. Now the problem is, over time, additional genes get turned on. So this would be the genes that are being activated in a nicotine-dependent organism, where even when they are exposed to the drug, you are not having as much of an effect. So then you have to consume more often to get the same effect. Addiction. That's how it kicks in. That's why these effects on nerve disruption and endocrine can be really important for developing tolerances and addiction. You also can have kind of the cell maintenance. This is the things that are going to keep you alive and going. So for example, protein synthesis can get negatively affected. In this case, we've got a toxin called Shiga toxin that we're increasing, and these are two different trials. And what we're looking at is the amount of isoleucine that is incorporated compared to a control amount. So we can radio label isoleucine and then track it going into protein. So basically 100% is no dysfunction. And then as we increase the toxin, we're seeing less isoleucine showing up in proteins, basically less protein being generated. You can also have energy pathways alteration. Again, remember the idea of messing with the electron transport chain in the mitochondria. And you can also have transport being blocked. So in this case, you have a toxin that is affecting the Golgi body, which is preventing the generation of lysosomes and the export of them. So you are not able to essentially generate external lysosomes to be able to process your food. So you can just mess with any of these systems. Now, I do want to hit the electron transport chain a little bit more because it can be affected very easily by toxins, and a number of toxins developed by organisms to kill other organisms work a lot on the ETC. Now remember, the ETC is in every one of your mitochondria, and it's its own independent organism, but you cannot survive without them. They generate 98% of your energy, so you are not able to survive without them operating fully. Now, different toxins are going to affect different parts of the chain. And remember that all the cytochromes, 1, 2, 3, 4, effectively are going to be pumping protons into that intramembrane space that then falls back through ATP synthase, and we generate lots of ATP from ADP, and we've got oxygen being used as your terminal electron acceptor. Different toxins can affect different parts of it. So for example, rotenone is a toxin that fisheries biologists love a lot, because one of the things that we'd like to do is we can use this toxin to stun fish. Now, in a fish, their cytochrome 1 can get blocked by rotenone. Their cytochrome 2 is still available. And remember, FADH2 can enter at cytochrome 2, NADH plus H plus enters at cytochrome 1. So effectively, we've cut off part of the electron transport chain, but not the whole thing. So the fish is producing some ATP, just not the full amount. And the cool thing from a fishery standpoint is as the organism starts detecting that they're running low on ATP, they interpret it as they're running low on oxygen, because fish normally when they're running low on energy, it's because the oxygen saturation in the water has gone down. So then they go to the surface to try to do RSV and get oxygen from the air by like gulping, if you've ever seen like your goldfish gulping or gulping in oxygen. And the response is you've got these tired, dazed fish now moving to the surface of the water, and we can scoop them up easy. Now, other things you can do, we've got some erythromycin, oligomycin. They're going to block different aspects of the chain and basically prevent it and back it up. Now, the other thing I do want you to appreciate is these ROSs, remember, reactive oxygen species. Anytime that you disrupt the movement of electrons and the movement of protons, they're gonna go somewhere. And in this case, if you're not having these protons get pumped, you can end up combining protons with electrons, making hydrogen, no big deal. But if you took that electron from another molecule, like a sulfated compound or a nitrated compound, you could make ROSs and OSs. So yes, we are poisoning the fish and doing some at least temporary damage when we daze them, but we don't do it enough that their antioxidants can't handle it. But if things get really bad and the whole system starts shutting down too much, you can have a secondary effect of those electrons building up on special proteins called caspase proteins that detect that the mitochondria is no longer working, and then it's going to go into apoptosis. Now, apoptosis is programmed cell death. It sounds like a bad thing, but it's actually a really good thing, because it's the idea of letting the cell kill itself in a very controlled form so that we can effectively take all of the components of the cell, break them up into little packages. Now, these packages are going to not be metabolically active, and they're going to be small enough that they can get absorbed by phagocytes for recycling or disposal. So if you look here, kind of a generic cartoon, we've got a cell, good old nucleus, some mitochondria, Golgi body, lysosomes, free proteins, things like that. When those caspase genes kick in and we start going into apoptosis, we're going to actually start having breakdown of those molecules and organelles. So for example, the nucleus is kind of losing its shape and it's going into pycnosis. Effectively, we're going to have the DNA break apart so that it's no longer active and form kind of clumps. And then it's going to fracture later into different little nuclear fractions through keraresis. Don't worry too much about pycnosis and keraresis in this class. But the thing to remember is we're going to form these blebs. And this is actually due to the shrinking of the cell membrane, forming little, what are going to become vesicles, little balloons. So we can see the blebs forming here on the left-hand side, where it's going to surround things like this mitochondria here, or these chunks of the Golgi here, and we're going to get little bags. Now, these bags are not going to be metabolically active, and they're going to be able to be eaten by your phagocytes in your immune system. Now, this is a good thing, because if the cell is damaged by a toxin or a foreign invader or something like that, if you shut it down, it's not going to continue to do damage. So for example, if this cell was affected by something that changed an enzyme, so now it's creating ROSs, those ROSs could then move to other cells in the local area and cause more damage. Or if it's a viral invader, it could essentially spread viruses after the replication phase. So destroy the cell before things get bad. That's the general idea. And do it in a way that doesn't spread the toxin or spread the invaders. That's the whole concept of apoptosis. Now, moving on to the next stage. Once we actually have all of our mechanisms being affected, we've gotten the cells, we've gotten the others, we have to think about then, is this permanent or is it going to be repairable? Now, if it's permanent, and the cells have now exploded, it's gone. We're going to just have to replace them by essentially rebuilding those tissues. But a lot of toxin damage is only temporary. So if the cells survive, you fix them. Proteins, break them down, recycle them. Yes, there are chaperone proteins that can help refold other proteins, like all of the heat shock proteins you may have learned about in like Dr. Samuelson's classes, but they're pretty rare for toxin damage. In a lot of cases, it's much better to just break them down, recycle them down to amino acids and rebuild them. Because remember, all you're doing is basically wasting energy. You're not wasting any of the building blocks that you can just reuse again and again. Now, you also, with lipids, can have chaperone proteins that can effectively return them to their original state. So if you had something like sulfated your phospholipids, so now your membranes are a little less fluid, like that medication I was talking about that caused the protons to leak through your membranes. Well, you can go through and just strip those sulfonations off. So again, just fix it using proteins. Now, even your DNA can get repaired. You've got enzymes to help repair your DNA by excising sections, replacing sections, making copies using your backup copy. You also remember that you've got the backup copy of your two sides of your of the double helix, so you can copy one side to another, built-in error correction, and you've got 2 copies of your genes, one from the mother, one from the father. And organisms like plants that are exposed to high levels of toxins, think radiation as a toxin, they're out there in the middle of a field, will even have higher counts of their genes to be able to do this repair via recombination. So wheat, living out in the middle of a field, bright sun all day long beating down on them, acute radiation sickness is what you guys would have. Sunburn that could lead toward carcinogenesis, skin cancer, not great. But they have to live in it all day long. Now, yes, they are able to absorb a lot of the damage by the fact that they're going to use some of that energy for photosynthesis, but still the UVs, especially UVA and UVB, that are in a wavelength where they can't absorb it, they're going to get damaged by it. So they're loaded with antioxidants to help deal with ROSs, and their genes, they got 8 copies of them. So if one gets damaged, they got backups. Now, the last thing for kind of repair at the cell level is to rebuild damaged organelles. If the system's broken, rebuild it. If the system is not repairable, go into apoptosis and kill the whole cell. Not worth trying to fix it if it's way too damaged. Now, a little bit on DNA repair. Your DNA can get copied, and you have that backup copy. So for example, you get exposed to UV light, you now have a damage where we've got an insertion mismatch. We can go through with small nuclear riboproteins, snip out the section, do an incision, excise the damaged section, and copy it again. This is happening all the time. You probably right now are fixing little errors in your DNA that has popped up. But the trick is, if the error doesn't get caught, or if the repair is too extensive, then you run into problems. But we do have built-in repair mechanisms. Now, jumping up to the tissue level for repair. At this point, you are able to start replacing missing tissues. If the cells are unrepairable, then you need to go ahead and just kill them with apoptosis. Again, it's a good thing. You can recycle the materials. You don't have to worry too much about the loss of things. You also will encourage more cell replication. Now, a lot of the times this will be produced by cells that are going through apoptosis, that they'll release a messenger that says, hey, other cells in the area, start replicating to replace me. You also can have, along with it, inflammation. Inflammation is a bad and a good thing. It's a good thing in that it will basically encourage more cell division, deliver more nutrients, also by increasing blood flow to the area, clear away and kind of distribute away toxin metabolites and other broken down bits. But it can cause essentially rampant cell division and scarification and things happening a little bit too fast. Now, your final outcomes from all of this is effectively you can have the tissues die, necrosis. Now, we worry a lot about necrotic tissue because when you have tissues that die, you can get secondary infections from things like funguses and bacteria. If you've ever heard of the idea of gangrene, and gangrene sets in and ends up causing a lot more damage. So, for example, thinking of a day like today, it's extremely cold. If you have your cells freeze and go necrotic, frostbite, that's really not too bad. You lose those cells, well, you replace them with some scar tissue via the process of fibrosis. But if you get a bacteria settling in, then you're in a lot worse shape, and you could get gangrene, and instead of losing a fingertip, lose your entire limb or your entire life when it goes systemic and its toxins go into your blood. So pretty nasty. Now, another common thing is fibrosis, scarring. This is the idea that when you repair, you try to repair the system as quickly as possible, basically filling in the holes. And you do it with non-metabolically active tissue. Well, it's not non-metabolically active, but it's different than. And it's not doing the same job as the tissue that it is replacing. So for example, you probably have scars somewhere in your body from a little cut. Those cells are not exactly the same as the surrounding skin. They tend to be more fibrous, which is why we call it fibrosis. Now, they're able to do some of the job of your skin, but not all. So for example, scar tissue a lot of times doesn't have the pores and the hair cells and other things. So it's able to plug the hole, but not able to do all of the jobs. Now think about this if this is something like liver damage or kidney damage. The scarring is not able to do the job of the liver or the kidney, and if enough of that builds up, organ failure. Now the last thing is carcinogenesis. And this is where we actually start having cell division kind of go off the rails, which is going to cause a lot of other issues. Now, we're going to get into a lot more detail of that a bit later. But for this lecture, this is where we're going to leave off. On Wednesday, assuming that they finish clearing everything up, we're going to jump into the next unit, which is going to be on the concept of risk and the idea of estimating risk. Now, don't forget, get your topics in. All you really need to do for those topic papers is your central question, the toxin that you are working on, and a little bit of a brief description on what you need, what it is going to do in the body. So, everybody stay safe, stay warm, and I will see you guys in a couple of days.